Okay, let's have a class. So, first of all, you need to read your tubes to see what the results looks like. So, 3 tube fermentation test. And read your tube. I would say if you have an E. coli, you will see a very obviously it is acid and gas positive. Is that correct? If you have E. coli. And if you have a CF, you should see it is alkaline. Alkaline. We won't say it's positive, it will be cherry red color. If you can see it, and if you see urea test, who has a citrobacter? What's the color looks like? Can I see the urea tube? Is that positive or negative?
Negative.
Yes. Whose urea tube is positive looks like? Change the color. Yeah, that was positive. Can I have it? What's the bacteria it is? E. coli, okay. It's not really E. coli, it's Serratia, to be honest. It's Serratia when you inoculate it, this urea test positive, looks like that. Cherry red. Okay, because phenol red at pH 14 is cherry red, so it's a Serratia actually. You inoculate the E. coli, but it's contaminated with Serratia. Okay, I'll put this back to you. So, urea test, I would say, is cherry red and positive. Okay. That's a pseudo E. coli, not really E. coli. Okay, how about the temperature? Does anything grow at 5 degrees Celsius? Should be nothing grow at 5 degrees Celsius if you look at that. Okay, no growth. And you should see everything, 25, 35 degrees Celsius, they should all grow. And how about 55 degrees Celsius? Only bacillus is growing there because bacillus is a thermophile. Okay, that should be the results, just to let you know. So that's some conclusion from the Tuesday. Okay, we're gonna move on to today. The lab biochemical test 2. We will do 5 tests today, so lots of work. Number 1, catalase test. Okay, catalase test, how to do it? Everybody grab agar plates. Agar plates, split them onto 2. Of course, you write your name, initial, and date there. On the left side, please use very gentle streak inoculation with Staphylococcus aureus. On the right side, please inoculate with E. coli, EC. We will do 35 degrees Celsius, 48-hour incubation. And what we're going to do then? We will add in H2O2. Because if bacteria has urease, have catalase, H2O2 will be breakdown, become oxygen and water. And oxygen comes out, usually means it's bubbling. And always remember, we talk about in the lecture, catalase test is to differentiate between Streptococcus versus Staphylococcus. It's an easiest quick test. Streptococcus is catalyst test negative, Streptococcus is positive. So that is the first test. You should see one of the side is bubbling, another of the side is not bubbling. On next week, Thursday. By the way, Tuesday there is no lab because we have a lecture, exam. Okay, I want everybody to do well, especially 9:30 lab. If I still have a lab, I have an exam, then you probably the focus is not there, to be honest. Okay, so this is a catalyst test. Number 2 is oxidase test. So oxidase test will be a similar method. You'll get another agar plates, split them onto 2. Write your batch number, initial, and date. On the left side, we will inoculate Neisseria. On the right side, we will inoculate E. coli. Okay, remember, we talk about Neisseria meningitis, Neisseria gonorrhea is a typical colony oxidase test positive, and the fastidious bacteria. Could be, okay, we'll see whether it grows. It should grow, maybe not. Looks like in a blood media, it grows okay. You see some sediment at that, but you have to shake it really well, okay? Now, what is catalyst test? This is we're going to talk about the electron transport chain. Very gentle, okay, very easy. You have FAD. Sorry, you have NAD, H2, go to FAD, then go to FES, FMN, and then you go to cytochroma, then you go to coenzyme Q, sorry, COQ. Then you go to cytochroma C, go to cytochroma A, then you go to A3, and finally is oxygen. If you remember, this is the electron transport chain. If the aerobic bacteria in the anaerobic respiration process, they definitely need to do, they're transferring energy. Now, when the electron transfer to cytochroma C, they need an enzyme to finish the process. That is cytochroma C oxidase. That is where this enzyme comes out. And how do we know it is positive? After 35 degrees Celsius, 48 hours, we will add is oxidase reagent. You don't have to know which one is that, okay? That is called oxidase reagent, a very long name. If you show purple color, it is positive. Other than that, it's negative. But always remember, oxidase test you have to do within a minute. Because there is oxygen existing in the air. If you wait too long, it's going to all become positive. Something I forgot about here. Catalyst test, never do on blood agar. Lots of the people in the clinical area, they think it's easy. I'm going to grow bacteria on blood agar. I can see hemolytic. Then at the same time, I can do catalyst test, but never should do it. Because on blood agar, naturally has oxygen. It will show you bubble anyway. Never do on the blood agar for catalyst test, okay? So second one, oxidase test. Third one, SIM test. Okay, SIM test is one shot 3 drops. And we will always say 1 step. So listen very carefully. For this one, we're not going to use a loop. We will using is a very sharpy, I would say like a needle. No loop at the top. And the SIM media looks like this. It's a semi-solid media. So you pick a bacteria, I would better say from the slant, go right in the middle, take it out. Only do that. Semi-solid media, that's all, okay? Why? This is a 0.5% agar semi-solid medium. Why do we want to make it semi-solid? Because we are looking for 3 things. Number 1, S indicates sulfide. What is I? I indicates indo. What is M? Indicate motility. So let's talk about these things first. The motility is very easy. If you see a bacteria, once you stab, you take it out, you see the bacteria growing everywhere. The whole tube is become turbidity, which means this bacteria has a strong moving capability. Now why? Because of the flagella, and the flagella is possibly cover the whole surface. Petrichals. Okay, if you see the bacteria only stay right in the middle, which means it is motility negative. Sulfide. Some of the bacteria could use sulfuric acids, then become what? H2S. And in this media, there is some of the metallic ingredients, like iron. And sulfide will make them turn black. So if you see the black color, somehow like this next week, which is sulfide positive. Okay, black color, sulfide positive. Now why bacteria has to use sulfide? They need going through anaerobic respiration. What is aerobic respiration? The final electron transport chain, final electron acceptor is oxygen. If the final electron acceptor is not oxygen, is other inorganic chemical, let's say sulfide, it is anaerobic respiration. Okay? Now let's talk about I. What is I? I is indo. So what is indo? It is a byproduct of tryptophan. Now the question is, what is tryptophan then? What is TRP? Do you still remember amino acid structure? You can't forget about it, okay? What do you have? Is that right? Tryptophan. Okay, bacteria can use tryptophan. If bacteria use tryptophan, they will cut right in the middle. What is this? Indo. Now, how do we know it is Indo? We inoculated. Next week, Thursday, we're going to add one big drop, a reagent called CoVac. A reagent, okay? One big drop. That means whether it is Indo or not. If it is cherry red on the top, it is Indo test positive. This test, we will do a assigned culture. Okay, we'll do the assigned culture for this one. Okay, so that's SIM test. Now, what is this guy here? Amino revised pyruvic acids. Is that right? They could go through TCA cycle or further aerobic respiration process. So bacteria use it, can use amino acids. Okay, that is Indo test. Number 3. Number 4. Phenylalanine test. Okay, how to do phenylalanine test? It's very simple. We have a slants right here. Phenylalanine slant. And we're going to inoculate you assigned bacteria. Use zigzag inoculation. And then, after 35 degrees Celsius, 48-hour incubation, we will add a reagent, which is FeCl2, ferric chloride. If you see a dark green color, it is positive. Now we go back to see what is phenylalanine. Okay. Talk about this one more time. What is this? Alanine, is that right? Now you should know, your exam 1 of biochemistry is amino acid, is that right? Okay. Alanine. What is a phenylalanine? 3 become 2. I'm going to add something there. This guy will be adding there. Is that right? Phenylalanine there. Then you add APHNEL. Phenylalanine. Okay. Phenylalanine test, we are testing what? Is an enzyme called deaminase. What the D means? Take it out. So, if bacteria can use phenylalanine, they will take this guy out, break down. What is this side? Phenylpyruvate acid. Modified pyruvic acids. Why it is important? We'll talk real quick. We have glucose. Goes glycolysis become pyruvate. Is that right? This is what? A TCA cycle. This is a modified phenylpyruvic acid. The same thing. They could go into further biochemical reaction. Okay? That's why it is difficult. It's do the research, do the test is easy. But you need to know the story behind that. Okay? Number 5, last one. MRVP test. This is a little bit of story behind it. What is MRVP test? This is the test. What we're going to differentiate is E. coli versus N. Enterobacter. Now, why we can differentiate these two bacteria? These two bacteria belonging a group called Enterobacteria ACA. They look very similar from much or many of the characteristics. It is gram-negative, large, non-endospore forming, so no endospore, lactose fermentation positive, gas fermentation positive, and aerobic bacteria, 2 facultative. So it looks very similar. Difficult to do the gram stain to test it. That's why we need to use MRVP test. And MRVP test is also a part of a group test called IMVC test. What is I? Right here, Indo. So this is Indo I. What is M? M is right here, is methyl red. This M is not motility. SIM, this M is motility, okay? This M is methyl red. What is V? Vogus Prescia. What is C? Citrate. Now, what is going to be the results looks like? E. coli is Indo positive, methyl red positive, Vogus Prescia test negative, citrate test negative. Enterobacter, Indo negative, methyl red negative, Vogus Prescia test positive, and citrate test positive. Do you think it's very funny? Looks like positive, positive, negative, 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 positive, positive, like a sticky ends of the DNA sequence. That's why this test is important. So you kind of feeling final exam you're gonna have get a little bit trouble. Is that right? Because a bunch of these things. So what you're gonna do today? Very simple. Everybody gonna get a 1 MRVP test tube. You inoculate E. coli. You bench partner inoculate Enterobacter. Okay? Now, let me make your life easy. Instead of you are struggling about the design of these things, SIM test, I want you do CF, you bench partner do EC. Is that okay? One person do EF and Citrobacter, another person do E. coli. Phenylalanin test, I want one person to do E. coli, another person to do Staphylococcus aureus. We'll see whether there's a difference. Is that okay? MRVP, one person do EC, another person do Enterobacter. Okay, is that clear? So let's go over it. Everybody got a place. Split them onto 2. Left side Staphylococcus aureus, right side E. coli for catalase test. It's need to do incubation, so the confirmation test will be next week Thursday. Oxidase test, left side Neisseria. Neisseria will be MP. In a blast, you need to shake very well, lots of at the bottom, okay? Right side E. coli. SIM test, you do Citrobacter, you bench partner do E. coli. Phenylalanin test, you do E. coli, you bench partner do Staphylococcus aureus. Last MRVP test, you do E. coli, you bench partner to do Enterobacter. Okay, let's make your life easy. Very important, no cross-contamination. If you cross-contaminated, everything looks the same. Another thing very important what you should do is a very good job of label. Otherwise, you're gonna on Thursday you will never find it. 2 plates, 3 slants, 1 tube, you're never gonna find it. I'm serious. So make sure you do a very good job of label. I would encourage you, label even on the edge a little bit. Put your initial on the edge, then you are faster to find it. Okay, write a star, write all kind of triangle to mark it by yourself. Okay, so that's what we have. We have lots of work to do.